I've been thinking, ever since you walked in, I've been thinking, this guy's English is ridiculously good for someone who's only spent 2 years in the UK.
Thank you. Thank you.
Hi, my name is Alexandru. I'm a DPhil student in molecular and cellular medicine at the University of Oxford, and I'm passionate about medical research on the diseases of old age. I've been fortunate enough to interview one of my favorite gerontologists growing up, and while it took me a really long time to edit this, I'm very happy to share it with you now.
Dr. Aubrey de Grey, thank you for having me.
My pleasure.
It's an honor. I wanted to start by saying, SENS is your famous proposal since the early 2000s, and its feasibility has been tested quite a few times in the past years. Can you tell us what are some recent achievements of the SENS Research Foundation that you are most proud of?
Things have been speeding up in terms of actual demonstrable progress. At SENS Research Foundation, we have always focused on the most difficult components of SENS because those are the components that other people are neglecting. You know, the pressures that exist both in academia and in industry are very strong to work on the low-hanging fruit, to do the thing that gets you results quickly. And that means that some things which are just intrinsically hard never get worked on. And we exist to fix that. What that means is, of course, that for several years we were publishing basically nothing, and it was very hard to demonstrate that we were making any progress at all. But in the past couple of years especially, that has completely changed. One huge breakthrough that happened about 18 months ago was when we were able to get our first publication in Science magazine, which was on our Crosslinking project. There had been for, I'm going to say, 15 years, an absolute logjam where nothing was happening in that field because the one problem that everyone knew was the central problem of the whole discipline couldn't be addressed. That problem was to analyze and work on this particular type of chemical structure that is called glucosapane, and which is the most abundant form of the cross-linking that is generated by the reaction of proteins with sugar in the circulation in the extracellular space. So the problem with glucosapane is that even though it's the most abundant element of the cross-linking problem, it's still not very abundant, which means that in order to isolate lots of it from tissue, from an old person for example, you have to do a very, very laborious procedure involving lots of enzymes, lots of different experiments, and you only get a tiny amount anyway. And that made it impractical to do lots of things that would normally be obvious if you didn't have that problem, like generating antibodies against the thing, or finding bacteria that could digest it, things like that. So what we funded this group at Yale University to do was to develop a method where you could just synthesize this stuff from small chemicals, from just regular, cheap, small molecules, rather than taking it from existing tissue. And that was something that, of course, wasn't our idea. Lots of people wanted to do that for many years, but nobody had succeeded. And we found the right group of people to fund, and they did it. It took only about 2 years to do it, and we were able to publish this in a very high-profile journal, so that was very nice. Then, more recently, we've also had our first successes in a couple of other very important areas. One thing that happened about 3 years ago was we were able to demonstrate that we could break down a particular type of amyloid that is very important in very old people, centenarians, people over the age of 100. Amyloid is something that most people know about because it exists in Alzheimer's disease, but most people don't know that there are other types of amyloid that exist in other parts of the body, not just the brain, and that also increase during aging. And it turns out that this particular one that we were focusing on in the heart is a massive, important killer of really old people, which means it's going to become an increasingly important killer as we get better at fixing the things that kill people at a younger age. So we were able to, or the group that we funded in a university in Texas, were able to identify a way to break down this protein using a special type of antibody, initially, of course, in vitro. It's very difficult to do that because the proteins that form amyloid are, of course, natural proteins that the body makes, and therefore they have an actual role. So you don't want to just blitz them. You don't want to just break them down indiscriminately. What you need to do is to specifically break down when that protein, when it gets into a misfolded state, a state where it's the wrong shape and it tends to stick to other copies of itself, and that's what amyloid is. And we were able to do that. Then just a few months ago, we had a huge breakthrough in our oldest project, the project that we've been working on for the longest time, more than 10 years. We had this great breakthrough in our longest standing project, which is the project to put backup copies of the mitochondrial DNA into the nucleus. This is something that, again, wasn't our idea originally. People have been trying to do this for 30 years or more, but they were getting nowhere. Basically, it was just too hard. And we were able to put together a bunch of different ideas and work, just do a lot of trial and error, and we have made a lot more progress. We haven't done the whole thing yet, but we're much closer to it than anybody else. In particular, there are basically 13 separate problems to solve because there are 13 proteins that are encoded in this special DNA that exists within the mitochondria, and we've got 2 of them working now, and we're pretty close to getting 2 more working. So things are going well.
You have made sound proof of concept last August when SRF researchers rescued human cells which had a mutation in the mitochondria by using nuclear gene therapy. And I wanted to ask what distinguished that research from the many others which failed the decades before.
Yeah, so this is a great example of the way research tends to go. Research almost always takes far longer than you expect it to because you think you understand the problem that you're working on and then you find new problems and so on. This was a classic case of that. When people started to think of the idea of putting backup copies of the mitochondrial DNA into the nucleus, they initially thought it might be fairly easy because there are 3 things that you have to get right, but those were all fairly understood. First thing you have to get right is you change the sequence in a particular way so that the cell outside of the mitochondrion will synthesize the correct protein. You have to change the DNA a little bit in order for that to happen because of the different machineries that exist in the 2 places. The second thing you have to do is put a sequence on the front of the protein that tells the cell that this is a mitochondrial protein, even though it's encoded in the nucleus. And the third thing you have to do is you have to make sure that when it gets into the cell, into the mitochondrion, it is suitably assembled and made use of as if it had been synthesized within the mitochondrion. Now, the thing is, all of these things by the mid-80s, when this was first discussed, these were well understood. People knew what the differences in the machinery were, so what changes to the sequence to make to compensate for the differences and to make sure that the correct protein was synthesized. They also knew a lot about what sequence to put on the front of the protein so as to make sure that the cell would direct it into the mitochondrion. And the third thing they also knew was that you didn't need to do anything to get assembly to work because the assembly machinery was all already encoded in the nucleus. And so, everything would just be there ready to roll when the protein arrived. But sure enough, it didn't turn out that way. At first it did. The first time anyone tried to do this, a group in Australia, they tried to do it in yeast, and they just chose 1 out of these proteins. It was a very small protein they looked at, which was only 40 odd amino acids, and it worked first time. They were able to demonstrate definitively that it really worked, that when the mitochondrial copy of this particular gene was broken, the cells were still able to do the thing they needed to do with their mitochondria because they were using this backup copy instead. But then when people tried to do the same thing for other proteins, for other genes, it didn't get anywhere. And after a few years, everyone gave up and decided, well, this is too hard. And we don't like giving up, so we didn't do that. Back in about 2000, I published a very elaborate and messy idea for getting around the problem that people had encountered. But luckily, we haven't had to implement that because better things have come along. So what is this problem that they encountered? The problem is something called hydrophobicity. Now, a protein is a large molecule, obviously, and in order to get it into mitochondria from the outside, you have to get it across a couple of membranes, which are the surface of the mitochondria. And one of those membranes, the one on the inner one, as it's called, has other jobs to do, which make it very difficult to get large molecules across it. So what basically has to happen is that the protein has to be pulled through, 1 amino acid at a time in a linear chain. Now, of course, all proteins are linear chains, so you might say, what's the problem? The problem is energetic. Some proteins have this property called hydrophobicity, which means that they don't like to unfold. They like to scrunch up into a big ball, and if they're in that state, you just can't get them into the mitochondria at all. So people found that all of the proteins that are naturally encoded in the mitochondria, the ones that we want to make backup copies of, had this property in spades. They really are very hydrophobic. So they just didn't work. It only worked for that one protein at the beginning of this whole thing 30 years ago, because it's so small that just by pulling really hard you could make it unfold even though it really didn't want to. It just didn't work for bigger ones. So then there was a huge breakthrough about 10 years ago which kind of simplified everything. The breakthrough was to identify a way of importing proteins into mitochondria while they are still being synthesized outside of the mitochondria. This is something that there had been hints that it should be possible because people would see this kind of thing happening in nature, and actually that had been discovered way back more than 40 years ago. But nobody knew how it worked or how to control it, how to use it on your protein of choice. And the group that figured this out found out that it was actually quite simple. You could essentially you could put an extra sequence on the back end of the gene rather than the front, which is what people were focusing on before, and that would help the protein to only be synthesized right on the outside surface of the mitochondria, and so that it could be imported while it was still being synthesized. This is important because the thing about hydrophobicity is that it really only happens to complete proteins. Essentially, you're not giving the protein the opportunity to fold up into this unimportable ball. Anyway, so we heard about this. I heard about this as soon as it was announced. And we actually put a PhD student into the lab in Paris that made this discovery for a short while. But eventually we decided we wanted to do our own thing, and we created a whole group to work on it ourselves. And we've combined a lot of other ideas, plus a lot of straightforward trial and error, of course. It's been really hard, but we're getting there now. We feel that we have pretty much figured out the main stumbling blocks that make it really hard for proteins to get imported. And we've got, what we published, as you mentioned, a few months ago, is that we've got 2 out of the 13 key proteins working. I can tell you that we have unpublished results that are promising, preliminary but promising, for 2 more proteins. So we're on our way. I think we're going to crack this now.
Does that mean 4 proteins in the same genetic fix?
No, this means 4 proteins with 3 genetic fixes. So it turned out that the 2 proteins, the 2 genes that we were in, what we were interested in at first, are overlapping in the mitochondrial DNA. So we could do 1 mutation and knock out both of those proteins. Now, in order to do the fixes, of course, we have to put both proteins separately into the nuclear DNA. And actually, yeah, we did that in a variety of different ways. You can put them on the same construct, you can put them on different constructs. It doesn't really matter. You know, what matters more is how you play with the other things, like when the protein gets synthesized and how much of it gets synthesized and all that kind of stuff. And so, you know, that's the easy part.
A lot of scientists have held resistance to SENS in the past. For example, a few scientists from the Bioinstitute of Aging have signed on papers which were criticizing SENS, while saying SENS is distracting from good gerontology research. Today, the Bioinstitute of Aging is collaborating with SENS on a new research. Even though you have great support from luminaries such as George Church at the beginning of SENS, there seems to be a growing support for it now as well. So, what are your thoughts on this?
I've certainly had to withstand a lot of opposition to these ideas over the past 15 or more years, and there still is opposition, but it's getting easier in a lot of ways. I think the best way to answer your question is to identify 3 different types of opposition, which have had 3 very different stories that have happened. The first type of opposition is not so much to the feasibility of this approach, but rather to its desirability. You know, people will often say, oh dear, it would be terrible if we didn't have aging, death gives meaning to life, where would we put all the people, you know, all that kind of thing. Now, that still happens, but there's less of it. People are getting a little bit more embarrassed to be saying such crazy things. People are beginning to understand that, you know, aging is bad for you, it causes suffering, and if we can fix it, that would be quite nice. So I'm kind of gradually winning that argument. But the non-scientists subset of humanity, the politicians, the general public. That's the main thing. They kind of don't really care whether it's possible or not, because they're so convinced that it would be a catastrophe, it would be a terribly bad idea, that they kind of anything about feasibility goes in one ear and out the other. So it's a problem. I mean, we've got to convince people more and more that this is a good thing, and it's gradually happening. So then there are two other types of opposition which exist in the scientific community. In the scientific community, people who study the biology of aging, there's hardly any of that problem with desirability. Almost everyone who studies aging understands that aging is really bad for you and that we really ought to fix it as much as we can, as soon as we can. Yes. But the question then is, how can we fix it? How difficult is it? How soon can we make how much progress? And when I first put SENS forward, I split it. Perhaps I wasn't explicit enough about this at the beginning, but I split it very strongly into two parts, chronologically. So let's call it SENS 1.0 and everything after that. SENS 1.0 consists of these 7 types of therapy to address 7 different types of damage that accumulate in the body during life and eventually contribute to the ill health of old age. And I propose particular implementations, particular ways to repair that damage or to make it harmless. And a lot of the technologies that I brought to bear on the problem, that I proposed as implementing this, were technologies that had been developed way outside of gerontology for completely different reasons, to treat childhood diseases, or in some cases not even biomedical. One big part of it was developed by people who wanted to clean up pollutants in the environment. A lot of different ideas that I brought together. And of course that meant that people who were focused on the biology of aging were being introduced to these ideas for the first time. And it's normal in science for people to start off being cautious and pessimistic and so on. A lot of my colleagues went too far that way, and they just didn't bother even reading my papers, let alone the literature that I was citing in my papers that would actually justify my statements. But, I kind of understood that was going to happen.
I would comment on that there was a paper I read about which, quote, journalists too often fall for the idea of a Cambridge scientist who knows how to help us live forever with telomerase, allotropic mitochondrial coated proteins, and marker-tagged toxins.
Yeah. So this is kind of the point I'm making, that I was probably careless in not distinguishing these two types of feasibility, SENS 1.0 and everything else. Because I never said that these 7 types of approach could make us completely eliminate aging and live indefinitely. I never thought that, and I certainly never said it. My feeling, and what I used to say right from the beginning, was that if we could do all of these things really well, with the kind of feasible technology that I believe we could implement in the next few decades, then the kind of amount of extra life we would get would be in the region of 30 years. I never said any more than that. And people kind of forgot that. I'll come to why they forgot it in a moment. But anyway, so the question then is, can we even get those 30 years anytime soon? And as I say, I came to that conclusion on the basis of bringing together a lot of ideas from other parts of biology. So sure enough, it took quite a few years of essentially me beating people over the head with this stuff and getting people to understand that this was real science that came from elsewhere and that this was all feasible. And it worked. It took a lot of effort, and early on in the process, in like 2004, 2005, I had to be quite vicious about it. I had to actually make people angry enough to say in print what they thought was wrong with these ideas, so that I could actually write cogent defenses of them, rebuttals of the criticisms. It all went extremely well. I was able very quickly to demonstrate that the skepticism was fundamentally based on ignorance, but it took time. And now, though, that's pretty much over. There are still some people who have so much emotional investment in their own ideas that they will continue to say that the whole thing is nonsense. But most people have totally come over to the view that maybe it'll work, maybe it'll take a bit longer, maybe Aubrey's a little bit over-optimistic, but the whole thing does make sense. And indeed, people are reinventing these ideas. Over the past few years, there have been a number of papers that have come out that have essentially described the same thing, pretending that it's an original idea and not citing me, of course, but that's fine, really. Because if they're saying the same thing, a divide and conquer, damage repair approach, then the result is the same. And these papers, unlike my original ones, are being very highly cited, very widely, referenced and everything. So it's become legitimate, it's become orthodox. But now, let's move on to what happens after SENS 2.0. The thing about SENS is that because it's repair, because it's rejuvenation, actually taking, not just slowing down the clock of aging, but actually turning it backwards, that means that you can buy a lot of time with it. You can take someone who's already in middle age, let's say 60 or 70 or maybe older, and fix them up well enough that, they'll only get another 30 years. They'll be biologically 60 again by the time they're chronologically 90. But that's 30 years that you've had to develop SENS 2.0. And then, of course, you can apply better the improved rejuvenation therapies to those 90-year-olds who are biologically 60 for the second time. If you applied the original therapies, you'd get nothing. But if you apply the better therapies, you're going to re-rejuvenate the same people so that they won't be biologically 60 for the third time until they're chronologically 150 or whatever.
That is the velocity.
This is what I called longevity escape velocity. Essentially, all we need to do is to improve the comprehensiveness of this damage repair approach fast enough to stay one step ahead of the problem, and then the same people will continue to benefit from increasingly high quality technologies. And if you ask the question, is it likely that we would be able to keep this thing rolling fast enough? The answer is it's as certain can be. The rate of progress that we would need is tiny compared to the rate of progress that we see in any other technologies in history, following, of course, the initial breakthrough that gets the whole thing rolling. We're talking about the subsequent stages where incremental refinements are being made. So it's completely 100% certain that this will happen. But, of course, the difficulty is that is what leads to these terribly scary numbers for how long people are probably going to live. And as you mentioned, gerontologists really don't like to be associated with statements that people who are alive today have a chance of living to 1,000. They run away very fast. But the thing is, though, there is absolutely no argument against this. Nobody has even tried to say why this is crazy. People focus on the near-term stuff, SENS 1.0, and the arguments for those, as I say, have more or less gone away. But then they'll still absolutely change the subject as quickly as possible if it comes to talking about SENS 2.0 or beyond. And I can understand that. These people have got to get funding from people who are terribly ignorant and stupid, right? And so they've got to be politically correct. I have been able to get myself into a position where I can afford to be politically incorrect. And of course the value of that is that I can attract support and money from people that my more conservative colleagues can't. Because there are people with serious money out there who are not interested in delaying aging by 10 or 30 years. They're interested in delaying it indefinitely and doing to aging what we have already done to polio. And so the more sensible and pragmatic of my colleagues recognize that the right thing to do publicly is not necessarily to agree with each other about these things, but not to disagree too strongly either. And so that's why you will see that there's very much less vocal, vigorous criticism of my long-term claims in print or in the media than there used to be.
Okay. You've stated in the book, And Engaging, that the first step towards better funding of the rejuvenation tech field is robust health span extension for mice. Is there any study combining the SENS strategies? And if in order to show the additive benefit, do you still believe, if there's none, in the need to show the additive benefit in a model organism, or would you rather just go for the
this is a great question. I'm still 100% convinced that the best and most effective way to really change society's attitude to this whole business of whether we can bring aging under medical control is to bring the expert scientists, the credentialed, acknowledged leaders of the field on board with being able to say in public, on camera, that, yes, we are going to be able, it's only a matter of time before we manage this. And I believe, knowing these scientists personally, as I do, that the one thing that will let them do that, that will make them feel that it's actually safer to be optimistic in public than to be pessimistic, which is not what scientists normally do, the one thing that's going to do that is to get results in the laboratory using the most studied mammal in the laboratory, which is the lab mouse, that are completely decisive. Now, what does decisive mean? I believe what it means is to have a large extension, maybe a doubling or trebling, of the remaining lifespan of mice that, A, have nothing wrong with them initially. They're not like aging in an accelerated way because of some mutation or anything. And B, that you've done nothing at all to them until they're already in middle age. Okay? There are plenty of ways to get mice to live at least a year longer than they normally would.
And
But
average lifespan is...
average lifespan is typically a couple of years, yeah, depending on the strain, but yeah, 1 year, 2 years, 3 years. There are no strains that naturally live on average more than about 3 years. Yeah, if you can like add a year to that, then that's interesting. But if you do it by some kind of intervention that is implemented throughout life, even starting when they're still growing, like, for example, feeding them not as much as they would like, that works, okay? And that was discovered in the 1930s, okay? That's not very interesting to people who are already getting on. You're already in their middle age or older. And it turns out that if you do those same things to mice, or indeed any other species, that are already old, if you start late, you get less of an effect, right? Essentially what you're doing with these things is you're slowing down the clock of aging. If, however, you can reverse the clock, you can repair the damage that the body's doing to itself, then it should work fine in animals that are already old by the time you start the therapy. And so that is going to be what's decisive. So I defined this goal that I called robust mouse rejuvenation, which essentially says you take mice that are already 2 years old, and they're a nice long-lived strain, so there's nothing wrong with them, and they would normally live another year on average. So hardly any of them have died yet, but they're all going to die within the next year and a half, and about half of them are going to die in the next year, okay? And you then do a bunch of stuff to them, and you treble that. You get them out to living 5 years total, rather than only 3 years total. I believe that if we can do that, game over. It will be impossible for gerontologists to continue to think that this is all a pipe dream, or to say that it's all a pipe dream. And as soon as gerontologists start saying, oh, that's correct.
Oh, gosh. I'll be right back.
Thank you.
Nothing.
You sure?
Thank you very much.
I'll be right back.
Thank you. So as soon as gerontologists start to say it, here's my thinking. Money mostly comes from the government. The government chooses to spend their money on the basis of one criterion above all, namely the likelihood that it will get them re-elected. So ultimately, government money follows public opinion. And public opinion, therefore, has to change. So, what does public opinion follow? It follows the opinion of opinion formers. Oprah Winfrey, people like that. What do they follow? They are very smart people. They got to be opinion formers by being very smart and by knowing when an idea, an opinion, is credible enough that it's going to be right, but knowing it, thank you, but knowing it before most people see it. So they can pretend to be in the lead when in fact they are following as well. Who do they follow? In practice, when it comes to anything, they follow the experts. So if Oprah Winfrey or someone or people like her start to see that the wind of change is happening, that experts in the biology of aging, not just one expert like me, but experts in general, are saying more optimistic things than they used to say, then people like that are going to get on board. And the effect, the knock-on effect on public opinion and on public policy is going to be very rapid. So I'm still convinced that that's the way to go. So then the question is, how do we get there? Now, it's a stretch goal, trebling the lifespan of a 2-year-old mouse. A mouse that's already 2 years old and has only got 1 year to live. I am still completely convinced that we will not do it with just one therapy. We will not do it in any organism, certainly not in mice, just by fixing one type of damage. We've got to be reasonably comprehensive. I think we can probably get away with being less comprehensive than we would have to be in humans, but we're talking about a proof of concept here, so that's okay. Still, we're going to have to do a bunch of different things in the same mice at the same time. Now, then the question is anyone trying to do that? And of course, there's a prerequisite to doing a bunch of things in the same mice at the same time, which is you have to develop the individual components. And that's the stage we're at today. We're still in the process of developing these individual components. But we're moving through that. In fact, now we're in a position where we can begin to actually combine things that are bona fide rejuvenation therapies. We could take mice, for example, and do certain stem cell treatments on them, and also do treatments that remove their senescent cells. Also, we could initially cheat. I mentioned that an absolute prerequisite here is to use mice that have had nothing done to them until they're already in middle age. But if we just do one thing to them in their germline, like do it to their parents instead of them, which allows us to do things later on more easily, then we're only kind of slightly cheating. And in particular, if the thing that we do is something that we know how to, we are optimistic that we will be able to do somatically later, then we've got a chance. So we've been thinking that way. A lot of the things that are going to need to be done will involve introducing new genes into the mice. That means gene therapy. Gene therapy is hard. At the moment, nobody can do it very well. We've developed mice that should be much more amenable to gene therapy than any mice that naturally exist. And we developed those mice in the cheating way I just mentioned, by messing around with their parents. But we know how to develop those mice somatically, by starting from mice that have had nothing done to them until they're already adults. And so it's kind of a first step. We're always looking for these incremental ways to put the pieces of the jigsaw together. So now I believe we are at the time where we should start the project of combining things that already work individually and seeing what happens. And I'm quite sure that the overall result eventually will be an achievement of robust mouse rejuvenation. Whether it takes 3 things or 5 things being done simultaneously remains to be seen. But until we try doing that, we're not going to know. And the thing is, of course, in order to do this at all, we need to escape from the normal constraints of research. Combining things that already work and showing that they still work, and they work additively or whatever, that's not scientifically interesting. It doesn't test a hypothesis, really. It's technologically interesting, but it's not scientifically interesting. And that, unfortunately, determines whether it's going to get published in Science or Nature or Cell. And that determines whether scientists are going to want to work on it or apply for money to work on it. So the money for this kind of thing has to come first. We have to attract funding that's visionary enough to understand all of this outside of the main system. If we've got the money, then the scientists that have the expertise to do the work will do it, whether or not they can get it published in top journals. And eventually, of course, they sure as hell will get it published in top journals because the results will be so dramatic, right? So it will actually happen, but it needs visionary money to come in first. And that's another reason why I go out there and I talk to visionaries, whereas most of my colleagues talk to the government.
So what do you think of Liz Parrish and people in the biotech companies that are pushing for faster developments through patient zero tactics? Do you feel they deserve the criticism they receive from the scientific community?
I'd like to answer that question by combining it into a more general answer, because the situation around the process by which a potential therapy moves into general use is subject to very much the same kind of constraints as the research process, where people choose what research to do and how they get money for it. Namely, there are lots of different constituencies with lots of different vested interests, and they don't necessarily work together in the most efficient way. In the case of regulation, in other words, the process of getting something out into the general public, the first problem is that regulation is something that the government does, just like research funding. And so the government, because they want to get re-elected, they want to make sure that they adopt a policy that is aligned with what the public think. The public tend to be very risk averse. When one person dies in a clinical trial, it makes headlines. When millions of people die because something took too long to be approved, it doesn't make headlines. So that's a real problem. But conversely, some people anyway, are beginning to recognize that it's a real problem, and they want more intelligent, more sophisticated approaches to the regulatory process that will allow drugs to get out there and save lives a bit sooner than they currently do. And so there's a lot of activity going on now, trying to introduce this greater sophistication, better follow-up, better analysis of data that comes from patients who are getting a drug that's already been approved, what's called phase 4, things like that. And it's happening all around the world. Some countries have different emphasis than others. For example, the FDA has been very, very focused on not only making sure a drug is safe, but also that it's better than any other drug that was previously approved. The European situation is a bit more flexible. They're mostly focused on safety. They are a bit looser when it comes to efficacy, relative efficacy. That may also be changing. There are definitely people who are potentially going to shake up the FDA in the US and make it more friendly towards drugs whose efficacy has not yet been shown, and to focus more on safety. But that remains to be seen. But these are really details. The main thing is we need more data and we need better use of data. And to come back to the question you actually asked, self-experimentation plays a big role in this. When people go out there and they try something experimental that's not approved yet, and they try it on themselves, then bad things will often happen. But good things may sometimes happen, and whichever happens, if it's all done in the public eye, we learn from it. The world learns from it. And so some people are more risk averse than others. I would not do what Liz Parrish is doing, and she knows that. But I am still pleased that there are people doing the kind of thing that Liz Parrish is doing. And that's why I give her scientific advice, and I'm publicly associated as a scientific advisor to her company. Self-experimentation has a long and distinguished history. Many, many famous scientists have engaged in it. I often cite the case of J.B.S. Haldane, who was a British geneticist back in the early part of the 20th century, one of the most famous and prominent biologists of his time. He was quite famous for doing quite a lot of self-experimentation. So it's not in itself wrong. And when you hear people condemning all of this, or for that matter condemning medical tourism, people going to countries with more relaxed legislative jurisdiction on regulation of drugs, to get drugs that have not been approved in a country like the US. You know, a lot of it should be taken with a pinch of salt. The people who say these things from a position of scientific authority are saying what they need to say in order to be responsible, but for some audiences they have to oversimplify what they really think, and they have to say things that are not very nuanced, you know, that are really overly conservative, more conservative than would really be reasonable. They are not slowing things down as much as you might think just by, you know, not reading between the lines.
Let us say there's a millionaire or even a billionaire viewer of this interview who wants to support the SENS Research Foundation. What would you tell them that your money could be used for, and what topics do you need most of the help?
There is a concept in the whole world of funding of enterprises that are eventually very profitable, which is called the Valley of Death. Every investor knows what the Valley of Death is. And the Valley of Death applies very, very strongly indeed to the problem that we're talking about today, the problem of developing therapies against aging. Let me tell you what the Valley of Death is. It's easy to get money from academia, from the government, from public sources, to find things out, to do basic science. When I say it's easy, what I mean is people have careers in it. It's certainly not easy enough. People have to spend far too much time writing more grant applications than they ought to have to write in order to get that money, but it happens. And people can carry on doing that kind of thing all their lives and retire feeling that they've had a good life. Similarly, if you've got something that's close to profitability, something that people are likely to want to buy and you have nearly developed it, so it might be on sale next year or the year after that, then it's also easy to get money. You can get money from people who want a slice of that, and they're willing to spend money on you to let you make that final step to get to the point where you will make money, so long as they get some portion of that money back at the end of the day. That's investment. The problem is that those two components, the beginning and the end, have a gap between them, which is called the valley of death, where things look to be too close to fruition to be really basic science anymore. So the government gets less interested and other public sources of funding get less interested. But equally, it's too far away from profitability, so investors aren't interested yet. And a lot of projects spend a lot of time stalled at that point. Now, in aging, we have what definitely will at some point in the future be by far the biggest industry that humanity has ever seen, because this is something that everybody wants more than they want anything else. But it has to get there. We have to actually have the products. And it's difficult. It's no accident that we've been trying to solve aging since the dawn of civilization and we haven't done it yet, right? So a lot of this stuff is still at an early stage. And we at SENS Research Foundation are the entity that is at the spearhead of bridging the valley of death in aging research. We do stuff that is early stage, and we seek to get it to a point of sufficient proof of concept that it will be taken on in the standard way by industry. But we can't do it on our own. We're very small. We only have about $4 million a year that we can spend, because that's all we have. That's all that we're given. So every dollar that comes to this foundation, to SENS Research Foundation, allows us to get a little bit further into the valley of death, across the valley of death, from the left-hand side, from the beginning, from the basic science side. Every dollar that goes into the investment side also can potentially bridge the valley of death from that side, from the right-hand side, if it goes into the earliest stage companies. And what we have been able to do in a few cases already, and we want to do it much more, is to bridge that gap completely. What we've done is, on the one hand, we've taken a project far enough along that it's getting interesting, and then also we and our colleagues have found investors who are visionary enough to be willing to take the long view, to recognize that there's so much money to be made in the end that it's okay to actually lose money for quite a long time. So we've been able to transition now 4 or 5 projects from the foundation into the private sector, and those companies are doing well. So that's very nice to hear. That's great. But there's a problem. There's still a big problem. Every time we succeed in doing that, we end up in a position where the visionary investor chooses to be a visionary investor rather than being a donor. We still need the non-profit, the foundation, to carry on being the engine room, taking the harder projects up to that proof of concept where they can be privatized. And that's huge. The first project that we privatized, which is our work on atherosclerosis that we started back in 2005, what happened was that someone who was giving us a lot of money, starting in, I think, 2010, in 2014, he decided he decided that he wanted to start a company using this project. And we were delighted. That was fantastic, because he was going to put more money into it, it was going to move faster and everything, and that's happening. But the downside was he doesn't give us any money anymore. So that one project is doing fine, but we also have a problem. So that means that what we need is investors who are really visionary, who on the one hand they want to make money, and we will show them how to make money, because not many other investors will be willing to get into the same things that will be competing with them to get into the same things that we are moving along. But also they need to be visionary enough to bridge the gap from the left-hand side, to help us to get those projects to proof of concept. So, of course, the classic example of an investor who really has done that consistently for a decade is Peter Thiel. He came in when we had no hope of getting anything anywhere near the private sector for years, and he started giving us in the region of $1 million a year back in 2006, 2007. Over time, he's become increasingly itchy about it, not making any money, but he's still giving us that. This year he's giving us $1 million, and he's doing it because he has seen our successes over the past few years in moving things through to profitability. All right, so cross-linking. So way back in 2000, in the summer of 2000, when I had this overnight eureka moment that, that damage repair could be comprehensive, this was a very big component of it. The reason I was able to put all that together that one night was because of a variety of things, some things that gerontologists kind of slightly knew, like stem cells exist, which is about as much as gerontologists knew about stem cells back then. But a lot of it was stuff that had been done outside of gerontology, and a lot of it was stuff that kind of, some people were on the periphery of gerontology. And cross-linking was one of those cases. There had been, people who worked on glycation, on non-enzymatic glycosylation, would occasionally come to aging conferences. And it had been since 1981 that the idea of glycation, of cross-linking being an important component of aging, had been talked about in print. But it was still pretty much a backwater. Most gerontologists didn't really understand it at all and didn't really understand why it might be important. So the key result that led me to be able to incorporate cross-linking into my grand scheme was only published, if I'm not mistaken, in 1996 by Tony Sarami's group. They showed that, they claimed that they could restore the elasticity of the extracellular matrix in rats using this one drug. And they talked about it as a rationally designed thing. They thought, well, this kind of cross-link probably exists, it's probably really abundant, this kind of molecule ought to break it. Complete nonsense. You never actually, when you read a scientific paper, you never actually understand what people actually did. What people actually did was they were looking for something that slowed down the creation of cross-links, and they completely accidentally found something that appeared to actually reverse cross-linking, and then they backfilled to come up with a hypothesis for why it might work. But the fact is, it did seem to work. And they privatized it rather quickly. They created this company, which went through a couple of different names, but Altion is the best name that was known. And Altion were able to demonstrate a number of results, not only in just taking tissue out of animals and saying how difficult it was to break it, but also actual in vivo work. And it looked really promising. They were looking especially at hypertension, which is, of course, a major consequence of cross-linking, because it reduces the elasticity of the major arteries, and that basically means that the heart has to pump harder in order to get blood around the whole system, because energy is lost. The results were very promising. They didn't pan out in clinical trials, and still nobody knows really why. But my best guess is still that it's because in really long-lived species, you get a different distribution, different relative abundance of different types of cross-link. This particular type of cross-link that the drug they had developed is supposed to break is a particularly fragile one, which probably breaks spontaneously all the time. Which means, of course, that it's going to reach some kind of equilibrium, steady state level of abundance, even though it may be being created quite rapidly. And something that's being created more slowly but is stable and just carried on accumulating and doesn't reach an equilibrium level, will eventually overtake. So maybe that's basically what's going on here. There are other theories, but yeah, the fact is it didn't really work. However, you ask an excellent question. Why don't vets use it? Because, as you rightly remember, it has been successfully reported as being quite effective in animals. I think there may be a combination of reasons. First reason may be simply that the company, which owns, of course, the intellectual property, went bankrupt. They basically hung their hat on clinical applications, and it didn't work out. So it may be difficult to get one's hands on the stuff legally. You may be infringing patents that are still owned by a company that isn't functioning. Second problem is that the conditions for which the drug was shown to be effective in certain animals may not be very interesting to most vets. Dogs, you can reduce their hypertension using this kind of thing, but is hypertension a major veterinary concern, for dog owners? Maybe not. Another part of it is just lack of knowledge, of course. That happens all the time. People just may not have heard of this stuff or may not think that it works as well as it was published to work. A lot of things don't. There may be a combination of reasons. But as things stand, it certainly makes sense to at least have a go. And actually, this comes back to one of your other questions. There are other treatments for aspects of aging that are being tried in dogs right now, and why not combine them? So for example, right now there's a study being coordinated out of the University of Washington in Seattle, looking at the effect of rapamycin on aging in dogs, and it's a very exciting study. It's getting a lot of attention. I would love to see a therapy that looks at rapamycin combined with something that is not just a CR mimetic, something that actually is supposed to repair things. However, one of the rather irritating aspects of the old 711 story is that it has kind of ended up pushing the whole cross-linking area even further out of the gerontological limelight than it was before. If you look at the reinventions of SENS that have been coming up over the past few years, then you will notice that there are some differences from the original version of SENS, and one of the most egregious cock-ups is that the word glycation just doesn't exist in, for example, the Hallmarks of Aging paper. They just completely forgot about it. It's just like, what? And, yeah, exactly. Yeah, you're quite right to look incredulous. This needs to change.
I wanted to ask what the progress is with finding an alternative that actually cleaves glucosepane, pentozyme.
So at the moment, we really only care about glucosepane. And the reason we only care about it is because nothing has been found that in human tissue appears to be anywhere near as abundant. So there may be, there are definitely many other crosslinks that accumulate with age in human tissue. But if they only accumulate at 1% of the rate that glucosepane accumulates, then who gives a damn, right? This is the whole point of longevity escape velocity, right? If you can fix the biggest problem, and if the problems just additively contribute to the same pathology problem, namely loss of elasticity, then you may as well just fix one component at a time. So we're only looking at glucosepane. That's not completely true. So we have this group at Yale who are looking at that, but we also have another group in England at what's called the Babraham Institute, which is associated with Cambridge. And what they're doing is looking very much more rigorously and systematically than anyone ever done before to see if there are other crosslinks that have not been identified yet. Think about chemistry, like science in general. To an extent, you find what you're looking for. And so it's very possible, and the more that these people look at the literature, the more they get scared of this, very possible that there are other crosslinks out there that are as abundant, or whose abundance increases as rapidly with aging as glucosepane does, or perhaps even more rapidly, and yet they haven't even been identified at all. So that really needs to be solved, and that's why we're putting money into it. But the group at Yale are focused on glucosepane. Having developed this fabulous method that creates the stuff, grams of it for pennies, it means we can do anything we want. And even though they are chemists, they love doing new stuff. David Spiegel, the professor there, is a fantastic person to work with from the point of view of me as an overseer and a funder and so on. He's so creative and so responsive, and he just loves doing new stuff. So he actually has done some immunology in other areas that were unrelated to any of this, and so he started to do that. He's been generating antibodies against glucosepane. Antibodies are really important. In principle, we could generate antibodies that would actually break glucosepane, but that may be very difficult. The antibodies that break proteins that we developed in Texas for this completely different reason to do with amyloid, they would have to be repurposed quite dramatically in order to break something that isn't a protein, right? Of course, glucosepane is a linker between proteins, but it's not itself a protein, so we have a bit of a problem there. However, the thing is that if you can create normal antibodies that just bind to the thing, then you've got a tool with which to be able to measure the effectiveness of something else that does break it. You can see whether the abundance is changing. And more than that, you can you can see, for example, in much more detail where the glucosapane is. Which proteins it tends most often to link between, things like that. And we need to know these things in order to develop rationally develop drugs. He's also doing some of the things to develop drugs. So one of the difficulties that has essentially diminished interest in the field in looking for ways to break glucosapane is people looked at the thermodynamics. They looked at the energy level in the molecule, and they basically said, well, okay, this thing at low pH, it breaks down quite easily. But at neutral pH, at physiological pH, it basically doesn't. It's rock solid. So that means that in order to break it, you would need a rather reactive molecule. And reactive molecules have side effects. So you would expect not to be able to develop something that could be therapeutically useful. Bit of a shame. However, that does not apply if you go to enzymes. Enzymes are mostly anyway, they are very, very, very specific. If you have an enzyme that binds to glucosapane and breaks it, it's not going to bind to anything else. Glucosapane is a very unusual structure with a 7-membered ring in it. It looks nothing like anything that the body makes on purpose. So the chances are very high that we could identify enzymes that were specific enough. The question is, can we identify enzymes at all that even slightly break the thing? And so what we're doing is very similar to, or what Spiegel is doing at Yale, is very similar to the work that we've done to break down waste products inside cells. We're looking for bacteria that are able to do this. And that turns out to be not so easy as you would think. When you break down a waste product, then you can break it down as much as you like and you can get energy from it. So it's quite easy to find bacteria that have just evolved to break down waste products in the environment because they live off them. Fine. And it doesn't necessarily need to be energy, it could be carbon or it could be nitrogen. Something that they just for whatever reason can't get from anywhere else, or they have competing other species of bacteria that are better at it and so they have to find some other niche, some other way to compete. You can't do that with a crosslink because the crosslink is a link between things. So if the bacterium wants to get energy or nitrogen for whatever from material that has glucosapane in it, it'll just break down the amino acids. It won't do the interesting thing that you want it to do. Long and short of it is, the people at Yale figured out an extremely ingenious way to get around this. Essentially, they're working with bacteria that have been mutated in such a way that there are certain essential nutrients that they can't make. And you can't just use any old essential nutrients. You've got to use ones where you can make that nutrient by breaking glucosapane, right? But not in any other way. That's the concept. It's a lot harder than it sounds, but they're doing some very clever stuff, and they seem to be getting some success.
What do you think about in silico strategies? Such as I've heard recently of a researcher called Alexander, who's working at in silico medicine.
You're talking about my great friend Alexander Zavoronkov, who in fact runs in silico medicine. Yes.
So what do you think of strategies such as machine learning, such as in silico generation of new peptides that never existed before?
Yeah. There's only really one question around that whole area, which is how quickly it's going to be useful. Absolutely nothing can be said that would argue that it will never be useful. It's definitely going to be very, very important. The question is how soon. Now, of course, different groups are getting different results, and they're, for the usual reasons, they're sometimes having to overstate the results. Alex's group at In Silico Medicine are doing really good stuff. I haven't been paying enough detail to the specifics to know how far along they are, how reliable their information is, but it's definitely going to be getting better all the time. And let me give you another example of a company that's doing the same kind of thing. So In Silico are mainly focused on the genome. The other company that I'm very excited about right now in this space is called BioAge. It's being run by an extraordinarily bright person who's also a close friend of mine, Kristen Fortney, who was previously a postdoc at Stanford. And they're mainly focused on the metabolome, so on small molecules in the circulation, using that in the same kinds of way, to get a better idea, a better composite marker of biological age and a predictor of what's going to happen to you soon and not going to happen to you less soon and so on. And they seem to be getting, again, very promising results, but of course, again, those results will get progressively more promising and more useful as time goes on. And that's going to happen rapidly. The fabulous thing about machine learning, about machine learning today, the deep learning the methods that have been developed, is that they seem to have done something that, of course, I was originally a computer scientist. From my background, I would have never believed it was possible. They found ways essentially to use enormous amounts of data in a way that extracts understanding, or at least the consequences of understanding, in other words, appropriate behavior, without a combinatorial explosion. It was always believed, and I certainly always believed, that the more data you were looking at, the more problems you would have getting any useful output, because it would just hit diminishing returns. You would end up confusing yourself more than anything else. And people have found ways around that. Outside of the biology sphere, I would say the most spectacular example is when last year a program was able to beat the world champion at the game of Go. That was, if you'd asked anybody 6 months beforehand how long it was going to be before that would happen, nobody in the expert community would have predicted a number less than 10 years.
Actually, I was excited about the poker one, which happened this year.
I don't know so much about...
Poker is so much more intuitive than chess or any other game where you just run a edge gradients and that's it.
I don't know so much about the poker result, but yes, it's also very impressive. I think the thing you have to bear in mind with that, though, is that they were using kind of the most restricted version of poker, the Texas Hold'em thing, where it's inherently, from an informatics point of view, even though it involves a dimension that Go doesn't have, namely probabilities and incomplete knowledge, nevertheless, it's not as complicated a problem. And so, I would put that below Go in the complexity stakes. I would say that's at the kind of level that the same company, DeepMind, were able to achieve the previous year, which was in video games. Getting the same program to learn how to play a couple of dozen different video games really well, better than humans, starting from zero, just from nothing. The thing about Go. And I have a bit of experience in this because I used to be I used to play Othello at international level, and so I actually know Demis Hassabis, the guy who runs DeepMind, from when he was an undergraduate. The thing about Go is that the way it was solved was completely different. The way you got to world-class performance, completely different from the way that was achieved for any other game, whether it's Othello, whether it's chess. In all of those cases, there was some machine learning involved, but most of what happened was just developing better understanding, especially chess. Chess, people just got better and better at encoding what a good move was. And to some extent with Othello, but overwhelmingly with Go, it was all a matter of the computer using trial and error and using it well, using it productively. That's an idea that most people have just completely given up on because it hit diminishing returns, as I say. And it was only really, I'm going to say, 5-7 years ago that began to change. Actually, again, it was started in the Bay Area by a professor named Jeff Hinton, who really pioneered new ways to do deep learning, and the results he got started to come to the attention of people who understood a bit of neuroscience. Demis Hassabis, actually, a bit like me, he had a combination career. He started off in video games and so on, and then he went and did a PhD in neuroscience, which was very successful, and then he's now combining the two.
Okay. I wanted to go a bit off topic right
Sure.
here. And I always thought that, so you are a former computer scientist. You got your PhD from the University of Cambridge under special regulations for dismissing the vicious cycle theory.
Well, the whole book, there was a lot. Basically, my first half dozen papers are in that book.
So you got your degree in the UK, and you left the UK and came to the Bay Area. So I always assumed, I might be wrong, correct me if I am, that you came here especially because of the density of visionary people that are around the place.
That is precisely why I ended up here. I didn't really come here deliberately, it kind of gravitated. But yes, it's precisely the density of visionaries, both wealthy visionaries who are in a position to enable this kind of work, and also the youngsters, the people who are actually willing to aim high and who want to actually make a contribution to the world's most important crusade.
So you've received funds from Peter Thiel, you received funds from Michael Grieve, and I always thought that an obvious place to pitch at in this area would be Google as well. So Google started a company called Silico with Cynthia Kenyon. Am I spelling the name right? She's a famous C. elegans researcher. She discovered genes that, when mutated, can, in a certain way, it can delay aging. Now, that is not a repair strategy or anything viable for people over their middle age. And I always wonder whether you ever pitched to Calico or Google itself. So people such as Sergey Brin and Larry Page.
So this is a long and, so far, very sad story. Larry and Sergey have been very integrated into the techno-visionary community of the Bay Area since the early days. Way back in like 2003, 2004, when I started getting a bit of attention and started speaking at conferences like that. That was when I met them. I met Sergey first in 2003, I think, and Larry in 2005. And, you know, even then, you know, they didn't have the kind of money they have now, but they weren't short of a few bob. So, you know, it was crazy to me that they were not actually on board immediately. This was, especially after like 2006, 2007, when Peter Thiel came on board. So Peter was really riding high back then. He was making a lot of money in his hedge fund and so on. You know, when he was publicly supporting us to a significant extent, it made no sense that other people like him would not be coming on board. And they knew each other. I mean, I met Larry at a dinner party hosted by Peter Thiel, right? So, I mean, it made no sense. And gradually I began to understand that, you know, every billionaire is different. And some billionaires are just really anti-charity. They just do not want to give money away under any circumstance. And I think Larry and Sergey are both that kind of person, really. They just think that the private sector works better. They don't disapprove of charity, they just don't think it works. And so I think that's part of it. But finally they got their shit together and they actually decided, well, now's the time to make a company to actually get this to work. And the good news, of course, is they have so much money that they don't need the company to make money this side of the next century, right? And they've said so.
And the Calico has $100 million in funding.
Don't even think about a number.
Even 0.001%.
Don't even think about a number. I mean, the rumor is that when Art Levinson, the CEO of Calico, was hired by Larry, he said to Larry, right, what's my budget? And Larry said, we'll tell you when we run out of money. So it is unbounded, all right?
Okay.
All right, so the question then is, what the hell are they doing with this money? Now, the first question you have to ask is, why did Larry and Sergey hire Art Levinson? I happen to know that they had decided a long time back that when they were going to do this, that was the man they wanted to run the company. And I was told confidentially about a year before Calico was announced that the person they wanted was vacillating about whether it was the right time to do such a thing, and that Larry and Sergey were of the opinion that if they couldn't get that person, they just weren't going to do it at all. They were that focused on getting Art Levinson. Okay? Now, you have to ask why. Well, there's one really good reason, namely Art Levinson is God in biotech. He created this fantastic company, Genentech, and everybody adores him. Everybody worships him correctly. You know, he's a fantastic manager. He knows how to do everything that you need to do to run a successful biotech company. But Calico isn't a regular biotech company, so you've got to think, well, how does that actually work? But maybe you could say, well, you know, Larry and Sergey are thinking long term. Eventually this is going to make them shitloads of money. And they want to put all the right structures in place to make it, capitalism friendly as well as doing the right work. And if Levinson reciprocally can come on board with the idea of doing a lot of stuff that is very long term, which is more long term than anything he would have done in the past, then no big deal. And that's what happened. So they decided to bring Art Levinson in. Great. And sure enough, Art did what you might expect. He kind of bifurcated the activities of the company into two parts. One part was relatively near-term commercial stuff, Genentech Mark II. So the headlines that you see from Calico, things like, we've done this massive deal with AbbVie, this other big pharma company. These things are what Art Levinson is known for. Here's meat and drink, no problem. And, great. And he's got other people like Hal Barron, who are from Genentech, who are perfect for this kind of thing. But then he's got the actual job to do, to fix aging, right? What does he do? Words fail me. The first person he hires to actually fix aging is an emeritus professor from Princeton who's in his 70s and who is on record as saying that he doesn't have a translational bone in his body. He is the most basic of basic researchers, David Botstein. And he puts him in charge of this thing as the chief science officer. Now, Botstein then does one thing that's sensible. He hires Cynthia Kenyon as his number two. Cynthia Kenyon is someone for whose both science and just advocacy I have enormous admiration. First of all, she made huge breakthroughs in the early 90s showing how to manipulate the longevity of short-lived organisms of nematode worms in the laboratory using simple techniques involving mutations of just one gene. And the second thing, and this is the thing I admire her for the most, is she went out and she said the kind of things that I say, but she said them a decade before I was saying them. She was going out there saying, listen, for fuck's sake, aging is bad for you, and we have an absolute duty to fix it, and we should regard this as a massive priority. And she was doing this from a position of genuine vulnerability. She was a professor at at UCSF already, but she didn't even have tenure back then. So she was very, very brave in doing all that. So, she doesn't have that basic science problem at all. She totally understands. She's totally mission driven. Unfortunately, she has a different problem, which is that the particular approach that she pioneered back in the early 90s to postpone aging and extend longevity in nematode worms doesn't scale. It works less well in mice than it does in worms. It works less well in dogs than it does in mice. It works far less well in monkeys than it works even in dogs. It's just not going to get hardly anything in humans, let alone humans who are already in middle age before you do anything. And she absolutely refuses to accept this. Even though there's overwhelming data showing this, as well as evolutionary theory, which is completely aligned with the data, nevertheless, she has actually said, on stage, sitting next to me, she has said that if you could do the same kind of truly thorough, truly systematic genetic screen in mice that you can do in C. elegans, that she did in the 90s, for longevity, then you would probably be able to get mice whose proportional increase in longevity is the same as what can be achieved in worms. In other words, you double, treble, quadruple the lifespan of mice. She actually said that, with a straight face, on stage, on camera. Now,
even if that happens, you can't do that. You can't do massive mutagenesis screens in mice.
Well, what I should have said at the time, but my jaw wasn't working because I was so astonished, what I should have said at the time was, well, Cynthia, I can't think of anybody better than you to write the grant application. And now, of course, she's got the money to do exactly that. And for all I know, because Calico don't tell you what they're actually doing, for all I know, that's exactly what she's doing. For sure as hell, if I was Cynthia Kenyon, that's what I'd be doing with an infinite amount of money. And you can. You actually could. With today's technology, you could do those kinds of screens. So, we'll know. But I know already that it's totally not going to work. And furthermore, she ought to know, and Botstein ought to know. So, what's actually been happening I don't know what the details, but yeah, I mean, Cynthia's probably got a whole bunch of stuff going on. She's probably spending shitloads of money doing sensible experiments that will lead to interesting but negative results, okay? They've also hired a lot of other people to do other stuff, but it's also totally basic science. You know, large, systematic experiments that will teach us stuff and not actually get us any closer to doing anything. And so I'm horrified. I think that there's only one explanation for how Larry and Sergey could have got into this position 3 years down the road with Calico, and that is ego. In other words, they've made their choice, they've made it very publicly, they've hung their hat on Art Levinson. Art Levinson has gone in a direction that is not going to give them what they want, but they have too much hubris to say so. Not to do anything, like fire David Botstein and put in someone like me, which is what they should have done at the beginning.
Do you think I should include this in the interview?
I've been pretty rude about Calico on camera before, so yeah, you can basically say all that. Maybe take out the swearing.
I've never seen like camera beeps during your interviews.
Oh, there are some. But yeah, I mean, we did pitch properly. So
I understand.
I met Art Levinson for the first time just after he'd been hired. So before much had happened at all, just after the announcement. I mean, it was too late. He had already hired Botstein, but he seemed very friendly and very receptive to what I was saying, and he knew what he didn't know. He knew that you know, he wasn't a gerontologist and that he had a lot to learn. I suggested to him that probably there were a lot of people who had told him to talk to me and a lot of other people who had told him not to talk to me, and he confirmed that yes, both of those things were true. But he was very good. And it took a while, but a couple of months later, I was able to coerce him into actually inviting me and a few colleagues from SENS to come to Calico when they still only had half a dozen people on staff. They didn't have a lab or anything yet. And to let us give a 2-hour presentation. And everyone was very polite. And Botstein didn't even fucking turn up. I was outraged. I got to see Botstein myself over coffee about a week later, and he basically talked about himself all the time. And it was like, so they're blind us off, yeah. But one day, they will see the light and start spending money properly.
I understand your outrage at Larry and Sergey, and I think they aren't justified in doing that if that's what happened. I can understand, for example, Bill Gates' point of view, where child disease is really important.
Right, Bill. I have absolute respect for Gates and for people like him, Buffett, for example. You know, these people have decided that what they want to do is to help the disadvantaged. And that's fine. I wouldn't take a penny away from what Gates is spending on that. But people in Silicon Valley well, actually Zuckerberg is more like Gates, I think. But most people down here, most of the wealthy billionaires down here, they aim high. They want to help the leading edge move forward, not just the trailing edge. And so it makes no sense. Larry Ellison, another fine example. So, of course, he started a non-profit, the Ellison Medical Foundation, in, I think, 1999. And over the 15 years that it existed, it spent nearly $500 million. And, I mean, why he started a non-profit in the first place is shrouded in mystery. Some people say that it was part of a deal with the IRS because he had a tax issue. But the fact is, he did it. And it could have made a huge difference. Right at the beginning, he decided to work on aging. He was advised to work on aging by a friend of his who happened to be a Nobel laureate, who was actually not a gerontologist, he was an immunologist, but he was a very old immunologist, and so he said, you should work on aging. So that was great, except for the next step, because it was exactly the same story. Ellison hired this guy, this immunologist named Joshua Lederberg, to be the chief of his scientific advisory board, and he got Lederberg to take the next step, to hire the person who would actually run the show. Exactly the same story as with Calico, right? And what Lederberg did? He went to NIA and hired the outgoing head of the biology of aging division at the government. Now, the stated mission of the Ellison Medical Foundation was to complement what the government is funding, right? To actually do stuff that wasn't being funded. If your mission is the exact opposite of that, and all you want to do is supplement what the government is doing, in other words, to just like add a couple of percent to the pay line, what do? You go and hire someone who's done that job for the government for the past 15 years. If you want to do, it would be impossible to find someone less appropriate to hire. And the guy ran the thing for 15 years, well, for nearly 15 years, and ran it exactly as one might expect. They did almost nothing of any significance whatsoever. They made no difference to aging research at all. The only thing that came out of it that was any use whatsoever was they gave David Spiegel, the Crosslinking guy, a very small amount of money to do a very small pilot project in this whole space. And that's how I found out about it. That's the one thing that they did that I can point to as having had any real significance.